Hello, my name is Kostat Singis, and I am here with Yingtong Guo to discuss our research conducted in Brown Lab, which concerns a genomic investigation of site-specific protein binding of ATR-dependent tandem repeats. So what does this mean exactly, and what does it entail? For context, we focus on DNA and the genome. During DNA replication, DNA polymerase stalling creates unstable sites with structures formed by single-stranded DNA folding. The checkpoint kinase ATR then comes to these sites and inhibits replication fork collapse and breakage of these structures. So, Brown Lab used ATR inhibition to identify over 500 of these sites in the mouse and human genomes that depend on ATR for stability. The lab found that these sites consisted of structure-forming repetitive DNA sequences. However, now we ask whether these ATR-dependent repetitive sequences have biological functions. Our mission is to use bioinformatics to investigate whether they play a role in protein binding, and to find any possible transcription factor binding sites. So how exactly did we do this? This is an example of a highly significant repetitive sequence from one of the sites. Repeats like this one were identified from ATR inhibition-treated mice by analysis in the Tandem Repeats database, which provided us with what the sequence was and how many times it was repeated in tandem. For each highly significant repeat, we generated its alignment data, seen to the left. This was used to find the most frequently conserved nucleotides in each repeat position-wise, or the consensus sequence, seen in blue. For instance, take this first position. Of the 10 times this sequence was repeated in tandem, we see that every time a thymine was in the first position, except 2 times which had cytosines. High frequency, like with the thymine, indicates that the nucleotide is well conserved, and low indicates the opposite. For further analyses, this alignment data was represented computationally through position weight matrices, where each entry is the probability of a nucleotide occurring at a position. These matrices were visualized graphically through sequence logos, as seen on the right. In the first position again, the thymine is more well conserved than the cytosine, as we saw before. Seeing which repeat regions are conserved over others is crucial in determining possible transcription factor binding sites. Now I'll pass it over to Yingtong, where she will continue with how we use this data to further our investigation.
Next, I'll talk about the two analyses we performed to find transcription factors. So for the bioconductor approach, which is analysis 1, I compared each consensus sequence to the position weight matrices of a whole bunch of transcription factor binding sites. The matches were then verified against alignment sequences of the corresponding DNA repeats. Then analysis 2, for the position weight matrix approach, Costa performed a stamp multiple alignment analysis through the FootprintDB metadatabase. This approach matched transcription factors to both conserved and unconserved sequence regions because it doesn't just take a consensus sequence into account, but also its alignments. So then Costa scored the conservation of transcription factors on motif similarity and e-value, which is a probability that we encounter a given match by chance when we search the database. Finally, Costa and I combined our results and selected from them the ones that also appeared in the ENCODE database, which showed us which TFBS repeat matches have been confirmed by other scientists. Then we also made sure that the binding sites and the repeats were both well conserved, which is the last step you see here. So after going through this entire procedure, we found that the TCT-GA etc. repeat on chromosome 11 overlaps with the binding site for GATA1, which is shown in this slide, which is part of a gene family mediating the development of red blood cells and platelets. We also found another binding site for GATA1 on chromosome 10, also this one, and one for TCF12 on chromosome 18, this next one. But these latter two are less exciting in a way because the repeats aren't as well conserved. We also found that both transcription factors bind to all alignments available for each of our 3 repeats, but we did have to work with a limited number of alignments. So that's the caveat. Taken together, the above evidence strongly suggests the existence of TFBS repeat overlaps, so our hypothesis is supported. In future, we will be searching for more repeats with TFBS that have available experimental data, and for those that don't, we're planning to perform some wet lab analysis to determine whether the proteins indeed bind to the repeats we selected via bioinformatic tools. Discovering the biological functions of these repeats would help us explain both their prevalence and their conservation in the mouse and human genomes. But For this research, we only used repeats from mice. So now we've reached the end of our presentation. Thank you, everyone. And in case you have any questions, you're welcome to contact Costa or myself.